The overall goal of this procedure is to create a planar gradient diffusion chamber to investigate chemotaxis in a 3D collagen matrix. This is accomplished by first fabricating a 3D silicone mold with the use of a live cell imaging chamber. The second step is to make hydrophilic and hydrophobic cover slips and to use them to assemble the 3D silicone mold. Next, the collagen cell mixture is added to the center compartment and allowed to polymerize. The final step is to remove the hydrophobic cover slips from the mold after incubation and to add plain media to one reservoir and media with chemoattractant to the other reservoir. Ultimately, confocal microscopy is used to track cell chemotaxis over time and visualize cell motion displacement within the collagen matrix.
The main advantage of this technique over existing techniques, like the bowed-in chamber and microfluidics, is it allows for a simple setup to investigate migration in 3D space with a simple diffusion gradient.
Using a disposable pipette, measure out 150 microliters of the aminosilane APTMS and pour it in a 50 milliliter tube. Then add 30 milliliters of 100% ethanol to the tube with a second disposable pipette and close the lid. Vortex the solution for 2 minutes to ensure complete mixing. Then pour the solution in a glass petri dish and set it aside. Next, pour 15 milliliters of 100% ethyl alcohol in a second petri dish and place it with the first dish. Using a new disposable pipette, measure out 30 milliliters of deionized water and pour the solution into